Hello, my name is Raiyaan Shaik and I'll be briefly presenting on the potential apoptotic effects of thymoquinone by P53 dependent pathways in MCF-7 breast cancer cells. Although advancements have been made in the treatment of breast cancer, it remains one of the most prominent causes of death, with approximately 276,000 expected cases in 2020. Current treatments include surgery, chemotherapy, hormonal therapy, biological therapy, radiation therapy. However, with these come drastic side effects. To avoid negative side effects, researchers have been experimenting with novel ways to treat cancer, including herbal therapy. One of the most prevalent herbal chemicals experimented with is thymoquinone, which originates from the Nigella sativa plant. It has shown to have many chemotherapeutic effects, including angiogenesis and upregulation of regulatory proteins. One of those regulatory proteins is tumor protein 53. P53 is a common gene which plays a role in the regulation of the cell cycle. In fact, 50% of all cancers have a correlation to the mutation in this gene. The P53 pathway is responsible for playing a unique role in conserving the stability in the genome by transcripting protective responsive to oncogenic stressors. As you see in this chart, there are many factors that may lead to the expression of P53. In normal cells, P53 binds to damaged DNA and promotes transcription of genes, leading to cell cycle arrest. P53 is negatively inhibited by MDM2. In the absence of these signaling factors, P53 is bound to MDM2, which stops it from binding to healthy DNA. However, DNA damage triggers the release of P53 and it binds to damaged DNA, leading to cell cycle arrest. P53 mutations occur due to alterations of amino acid sequences. These mutations create non-functional P53. The P53 mutation has been studied herbally in multiple research projects. The upregulation of P53 through thymoquinone has been seen in lung cancer and breast cancer. However, scientific literature has stated that these research projects have a lack of clear understanding of thymoquinone's molecular mechanisms, especially in breast cancer. The goal of this research project is to understand P53-mediated apoptotic pathway and thymoquinone's effects on MCF-7. In the literature, proliferative effects have never been done alongside apoptotic effects to see the relationship, so that is one aspect I will cover. It is hypothesized that a sufficient amount and treatment of thymoquinone in MCF-7 cells will lead to apoptosis through the upregulation of functional P53. This chart on the right best summarizes my thought process. MCF-7 cells prior treatment have mutant P53, which leads to a lack of protective genes being transcribed and no apoptosis. However, with thymoquinone treatment, the amount of functional P53 will overcome the amount of mutant P53 and restore the proper functions of P53. There were some assumptions made in developing the basis of this research study. One of them was assuming that the treatment of thymoquinone on MCF-7 breast cancer cells will have a similar chemotherapeutic effect as it had in the literature with lung and colon cancer. Furthermore, another assumption was that the treatment of thymoquinone on MCF-7 will mimic an apoptotic effect as it had in the literature with breast cancer. However, two aspects where my study will differ from the literature is that, for one, it will compare apoptotic and proliferative effects side by side, as well as to establish the effects for a wide range of concentrations rather than just for one concentration set for a prolonged period of time. Now I'll be talking about the materials and equipments utilized in this research. The breast cancer cell line MCF-7 was purchased from ATCC. The compounds utilized were thymoquinone, RPMI 1460 media, 0.25 trypsin, ethanol, cell titer 96 reagent, propidium iodide, PBS, Triton X, 1% penstrep, FBS, and an XNV. The pieces of equipment utilized were the XNV apoptosis detection kit, Indirect ELISA kit, real-time PCR kit, cell viability analyzer, 96 well plate absorbance reader, and the flow cytometry kit. The first assay used in the experiment was cell proliferation assay, and it utilized the 96 well plate reader. This is a colorimetric method to determine cellular activity in cells after treatment. The higher the absorbance, the higher the cellular activity. The second assay was the cell viability analyzer, and that used a VI cell reader to determine the cellular viability after treatment rather than just the cellular activity, to determine if the treatment was just hampering the cellular activity or actually killing the cells. Next, an ELISA is another colorimetric method of determining the amount of presence of P53 which was induced in the treated cells. This was done to test whether the treated MCF-7 cells would have a higher P53 expression than the non-treated cancer ones. Now I'll be going over the stages of procedure and methodology throughout the experiment. This research was divided up into 4 stages, including future research. Stage 1 of this research had a goal to understand the proliferative effects of thymoquinone on MCF-7. To do this, a baseline of thymoquinone concentrations was established from prior literature. The first range of concentrations was from 25 to 400 micromolar. This established concrete evidence of decreased proliferation. However, to better understand the effects, the concentration range was shortened. The expected result of stage 1 would be to see a trend line of decreased proliferation of the cells treated with thymoquinone as the doses increase in concentration. These are the specifics of the methodology that was used in this stage of the research. Moving on, the goal of stage 2 of this research is to analyze the apoptotic effects of thymoquinone treatment on the MCF-7 cells, as well as develop a better understanding of thymoquinone's effect on the P53 regulation using an indirect ELISA. Overall, the goal is to determine whether allowing more functional P53 to be present in the cell due to thymoquinone treatment will lead to apoptosis. The following slides are specifics of the methodology which was summarized earlier that was used throughout this stage of the research. Moving on, the goal of stage 3 of this research study is to analyze the effects of angiostensin, which is an MDM2 inhibitor, to see if there is proliferative or apoptotic effects on MCF-7 cells. This is part of my future research plan. To understand the proliferative and apoptotic effects of angiostensin on the P53 inhibitor MDM2, similar steps would have to be taken from stages 1 and 2. A base concentration would have to be developed through prior literature and gradually decrease the concentration to find the best set to further study viability. The proliferative and apoptotic effects would have to be analyzed side by side. To understand what angiostensin does to MDM2, an indirect ELISA would have to be carried out using the lysates of treated cells with angiostensin, similar to the lysates used with treated thymoquinone. Furthermore, once the proliferative and apoptotic effects as well as the ELISA data was established, a cell cycle analysis kit would be used to further develop the understanding of what exactly goes on when apoptosis is occurring. The following slides are specifics of this stage of research. Next, stage 4 of this research is focusing on providing a combinatorial treatment of both angiostensin and thymoquinone. The goal of the future research is to see whether a combinatorial effect will have a more profound effect than both treatments by themselves, and this has never been shown in the literature. I'll be going over the data found in these experiments. This first graph demonstrates thymoquinone's proliferative effects on MCF-7. The dosage range is 25 to 400 micromolar. As you can see, initially there is a downward trend in cellular proliferation. However, the trend stops, continuing and starts flattening out, demonstrating that the effect on proliferation has a limit. Figure 2 also represents the proliferative effects on thymoquinone. However, here the doses have been decreased. This has also been done in Figure 3 to even smaller set of concentrations from 2.5 to 25 micromolar. This was the data set which was used to study further viability. This figure demonstrates the viability scatter plot done from the set 3 of the concentrations. As you can see, the viability drops to a little above 80% of the control, which is very similar to the proliferative effects, which demonstrates a unique point that the proliferative and apoptotic effects of thymoquinone on MCF-7 cells have a similar extent. This figure shows an ELISA data of the upregulation of P53 due to thymoquinone. As you can see, at 25 micromolar, the upregulation of P53 is at 600%, which means that the proliferative and apoptotic effects due to thymoquinone treatment can be used as a reason for these effects due to the P53 upregulation. Now I can go over a quick analysis of the data. Figures 1 to 5 demonstrate MCF-7 treated with various thymoquinone concentrations, followed by measurements of proliferation and viability. Thymoquinone demonstrated to be effective at hampering cancer cell activity as well as decreasing viability. It also has determined that there is a similar extent to apoptotic and proliferative effects on the cells. The ELISA data is significant as it demonstrates the apoptotic and proliferative effects of thymoquinone is done through the upregulation of functional P53. Through this research, thymoquinone, just like in prior research, has demonstrated suppressed cellular proliferation and decreased cell viability. As the study went along, the concentrations of thymoquinone were decreased to better understand the effects. Overall, thymoquinone has shown to decrease cellular proliferation and viability, and this can be attributed to the upregulation of P53 found in the ELISA data. In the future, I want to follow up this research with the combinatorial treatment. Angiostensin is an MDM2 inhibitor. MDM2 is an inhibitor of P53, meaning that if concentrations of angiostensin are given to MCF-7 cells, the expression of MDM2 should decrease, and this would mean that hopefully the regulation of functional P53 would increase. Thus, a combinatorial treatment would allow for an even more effective treatment in the cancer cells. Hopefully one day this research can provide a basis for herbal treatments to cancer and enhance research knowledge on P53. Here are my citations. Thank you so much for listening.